I can start anyway. Project pretty well. Small room. Pilgrim Jackson, co-founder of YatiriBio, biophysicist by training, and went straight from my postdoc to Celgene, where I had 13 years of pharma experience in the R&D space. It was relatively obvious from the beginning that there's some issues with the pharmaceutical drug development pipeline. Guy was talking about them. A number of the other talks have talked about it. It's really not that efficient. That doesn't mean it's easy to solve.
Ready? Okay, now I can talk a little more quietly. And that doesn't mean it's easy to solve. Lots of people have tried, certainly. But one of the things we have to do is really take a step back and see where the issues are. Again, Kai brought up that 4% success rate. Like, think about that, 4%. So the $2 to $3 billion, it's gone up a little bit, Kai, I think now, cost for drug discovery, it really leans on that failure rate. So that's where we want to step in, and that's where we think innovation can have the biggest impact. And while I would love to have reasoning, and when Kai's system is fully ready, I do believe in some form of reasoning. We're using more machine learning. So to address that gap, the 3 pillars that we've built that I think are very important is that we work off of patient samples. A lot of the work that's been done in the past has been done on cell lines immortalized for decades and sitting in plastic. It's moved pretty far from the reality of a biological system. We believe in proteomics. It's really proximal to the biology. So there's been some great technologies for transcriptomics and genomics. They've pushed the field forward a lot. There's still a gap, and I'll show you a bit about that. So that's the first pillar, is proteomics on patient samples as a data set. Very important. Next step is you have to do something with this omics. And when there are rational ways to use it, we'll do that. But for now, we use machine learning. Correlative analysis is very important to pull all of this together. One of the biggest gaps across the industry is the waste of data. It's much easier to generate data now than it is to actually use it. And then the last step is we don't believe that we know enough about biology yet to do full in silico models. We have to have something empirical that we can test ex vivo. What we're using mostly is patient-derived models, organoids, stem progenitor cells, things like that. Overall concept is very straightforward. Oncology, if you treat oncology, which is what we focus on, but it applies to other diseases as well. If you treat oncology as a tissue of origin disease, pancreatic cancer, stomach cancer, what you're actually treating is a collection, a heterogeneous biochemical collection of diseases. So it's not a surprise that you take your rational drug design forward, that it often fails. It may be very good for a few of those patients. It may have tox effects on some of those patients. You don't get above the statistical significance in your clinical trials. If you can separate out a smaller biochemical subset, you have much better likelihood of a success. You have the ability of repurposing drugs that may be failed or are in a different indication. And that's what we want to do. I'm not going to spend a lot of time on this, but the end message, proteins are what drive the biology and disease. Most of what's done right now is measuring this. Illumina's done a fantastic job of making this very accessible to research. But the gap between this and the proteins, between the RNA and the protein, quite significant. These are a few examples of clinical studies across hundreds of patients where median gene level or RNA level versus protein level is actually quite poorly correlated. All that to say, we really need to start layering in the proteomics data as well as the RNA data. What we do is we integrate patient metadata, patient-derived cell sensitivity. So these patient samples that we have ex vivo systems treat across a number of standard of care drugs to get the perturbations, and then patient proteomics. That's the input. AI/ML models built, and a couple of the scientists are here. You can chat with them later. That know it much better than I do. And then we end up with models that are predictive of patient outcome. And this, we think, is really going to push the industry forward. The long-term vision is that oncology will be treated as a collection of rare diseases, and we'll have models for each of those diseases to match patients to the therapeutics that need them, and vice versa, therapeutics to the group of patients that will benefit. We have a fabulous team, and it's all about the team. Afshin and Mahan were at Celgene when I joined, so we worked together for 13 years. I went to graduate school with Raman. That's the founding team. We have deep pharma experience. Greg Reyes ran the oncology division at Biogen IDEC before he was site head for Celgene San Diego. Deep experience across financial side and the IP side as well. Scientific team is fantastic. Our head of proteomics was running proteomics core at MIT before he joined us. Salva, who's here, has been doing bioinformatics on proteomics for decades. It's a niche space that really does require a specific skill set. And most recently, he was in John Yates's lab as a staff scientist. And John Yates is the forefather of global proteomics. Jill Mesroff, deep machine learning knowledge. She came from the Broad before joining UCSD. And then we think it's really important that you go all the way to the clinic. Our first two models are ovarian cancer. A local gynecologic oncologist works with us for that. And AML, which is the most advanced. And Derek Steyerwalt has been a fantastic advisor. He's at the Fred Hutch with a lot of experience in the AML space. Which is why I came late, and why I'll be leaving early is to go back to ASH, which is ongoing right now. So how do we protect it? The data itself is probably the biggest moat. It takes a really long time to accrue fresh patient samples, run the proteomics at a high quality level. Patient-derived cells are using unique conditions that we've developed. And then the algorithm itself that produces the predictive models. We have repurposing possibility with terms agreed for one contract that has $30 million in milestone payments. A market expansion possibility that I'll show you with a conservative estimate of $250 million in NPV. Do have a services arm because both proteomics and the bioinformatics that we use on that is actually very cutting edge. And so we work with small biotech through top 5 pharma to do that. That extends our runway, but it also opens the avenue for the larger partnerships. That is what our main goal is. And then in addition to that, that same data set actually provides novel therapeutic targets because we have real proteome data on real patients that we can query in a number of different ways. We have a really nice collaboration with a biotech out of New York that's designing mini proteins using machine learning called Ordeos Bio for the AML project right now. All right, so we kind of talked about this, and I'm talking too much. So this is definitely something that should be talked about afterwards, and we can talk in more detail. But we bring in the data, we bring in our own novel models that we're talking about. Definitely pull in a lot of public data as well as commercial. Those cell lines that have been immortalized for decades, they still have useful data to give. We combine the sensitivity ex vivo, like we were talking about, with the proteomics to build this model. And what you end up in your UMAP space is pockets where a model might be sensitive, and patients nearby, you can start to say, okay, these patients will be sensitive to drug X. They might be insensitive over here, or maybe this is a combination play where that sensitivity, that cell line is sensitive to a combination. You can start to explore a lot more deeply, mixing both the empirical and the data side. So, is there a minute you want me to end before questions? Then I have questions after that.
Ideally, your questions would come before that ends.
Okay, well, we'll try and go really quick. Astellas has a compound that's approved for FLT3 mutant relapse refractory AML. We ran it through our models, and we found the positive case and a lot of the FLT3 wild type negative case where it doesn't work. But very interestingly, we found another population, FLT3 wild type, sensitive to gilteritinib. So this is that example where net present value with great gross margins for Astellas. This is one of those win-win situations where hopefully we can expand their market into the FLT3 wild type patients. This is the actual data that built that, and in the interest of time, I'm not going to go over it, but we can definitely talk about it with anybody that wants to talk about it in the future. Models built, not only this is the gilteritinib Astellas example, another big blockbuster drug here has a clear proteomic signature that's not currently being used. And so that's it. We have raised 1.7 million to start the company, opened the lab 21. Kind of spring of 21. That is our accomplishments here. We have a $3 million note open now, 1.1 million of it's closed. And then we're socializing now the $15 million Series A that we hope to do mid to late next year. And thank you for listening.
Just curious, how much of your own
Yeah, I'm not going to give we have trillion data points into our model. No. Yeah, we have 100 AML patient samples, and for each we quantitate 8 to 10,000 proteins. So for AML, that's that. And then we have currently exclusive rights to another 150 patients in AML. And then I mean, that would be in the hundreds of thousands you can pull. But for AML itself, there's about 400. There's proteomics data on about 400 patient samples. Quality varies, and a lot of what the early work is figuring out how to merge these data sets. And that's where some of the fun computation comes in. You go from individual protein levels to a functional space to actually do the merging across data sets. And I think we have like 50 ovarian cancer samples. That model is going to be commercialized early next year.
Pilgrim, how much correlation are you seeing from your patient-derived samples compared to the organoid data you're picking up? Is that lining up? I know that's
yeah, and there's backup slides and all of that, and we'll talk about it. But it's really interesting. You see huge heterogeneity across patients at the proteome level, of course, as you would expect, AML, heterogeneous disease. And then in the case of AML, it's HSPCs, hematopoietic stem progenitor cells derived from a patient and expanded for 2 weeks. We see quite tight correlation to the patient. And when you go to the pathway level, we see very good correlation to the patients at this point.
Thank you.
Just out of curiosity, what's your differentiating factor compared to SEER, Prognomic, Interven, Nautilus? I mean, these are all the proteomic space.
Yeah.
Right? Different flavors. And then just in the same pitch as well, like in the cancer space. So just curious, out of curiosity.
Yeah, absolutely. I don't consider SEER, Nautilus, Encodia, Quantum SI, Arisian, I don't consider those competitors. I would love them to succeed and put that data into our model. So the key, the thing we're doing differently is that we're integrating the ex vivo model systems that are patient-derived with the proteomics data. Currently, the gold standard is LCMS, as you talked about, is LCMS-based proteomics, and that's what we use. But as soon as any of those work at the throughput they claim and at the quality they claim, we would love to incorporate that data.
Any other questions?
Yeah, thank you. You mentioned that you have 84% of accuracy, including the proteomic.
Yeah, that was.
Okay.
So what it was is we did our prediction and then the internal accuracy on the data set. That particular one was a public data set because we're publishing it. The internal accuracy on that was 95%. And then we took our own patient samples, completely separate, run on our own machine, so a different proteomics machine than was there, and our own sensitivity test, and we got the 84%
84%,
accuracy.
Okay, thank you so much, Pilgrim.